This is chapter 9, where we're going to be looking at molecular biology. This is one of the more challenging sections for people a lot of times. If this is the first time you've heard this material, make sure to plan on having time to go back and review it multiple times. Look at small sections of it rather than trying to understand all of it all at once. So just a kind of an idea of where molecular biology is led to. This is Dolly the sheep. This was the first cloned animal, and so this was a big achievement in molecular biology to actually be able to clone an animal. Not sure how familiar you are with this, but we were pretty happy that we were able to successfully clone this animal, but of course what we knew was just the tip of the iceberg, so the cloning did not end up going as well. We learned a lot through the process, but that there is just a lot more than just the genetic information to making a cell. Some of the pioneering scientists in this field were Watson and Crick. James Watson and Francis Crick are pictured here with the American geneticist Macklin McCarty. They are the ones that are responsible for discovering the structure of DNA. We have Rosalind Franklin, who discovered the X-ray diffraction pattern of DNA, which also helped to elucidate its double helix structure. So the structure of DNA is going to be made up of a sugar, which is ribose, or deoxyribose in the case of DNA, it's ribose in the case of RNA, a phosphate group, and a base. Cytosine and thymine are pyrimidines, and guanine and adenine are purines. So these are your purines. Purines have a double ring. The pyrimidines just have a single ring. One of the ways to remember which ones are purines is the mnemonic pure as gold. So you look at pure for purines, adenine, and guanine. All the others are going to be pyrimidines. Here you've got your phosphate, your sugar, and your base. So DNA is going to form a double-stranded helix, which you can see here in the picture on the left. On the sides, in the light blue, these are your sugar phosphate backbone. And then the colors that go across like the rungs of the ladder, these are going to be your bases. So here you have adenine that's going to pair with thymine, and cytosine will pair with guanine. This is showing the molecular structure of your bases that are pairing here in the center. What we typically will see is you'll represent them with a shape where you're going to have guanine and cytosine. The guanine and cytosine fit together like a lock and key. They're complementary shapes. Adenine and thymine are complementary shapes as well. You cannot fit adenine with anything else other than the thymine. Here on the sides, this is the molecular structure of the sugar phosphate backbone. So we have RNA and DNA. RNA is ribonucleic acid, and it's going to have the sugar ribose in there. DNA is deoxyribonucleic acid. It has the sugar deoxyribose. The difference is found right here on carbon number 2. This one has a hydroxide. This one here just has a hydrogen. So this genetic information is going to be found in the nucleus of a eukaryotic cell. It's in the nucleoid region of your prokaryotic cells. So this is where your chromosome will be found. The chromosome is just the condensed form of the genetic material. So here in this cell, it's loosely coiled, and so we call it chromatin. So when we go about making chromosomes, we start out with this double-stranded helix that's the DNA. The DNA is going to get wrapped around these histone proteins. As it gets wrapped around, it's going to form these nucleosomes, which will be like little beads that are wrapped into chains. This will undergo further condensation and become wrapped up even more to make your chromatin. And then the chromatin will undergo further condensation to make your actual chromosome. What this is showing here is a duplicated chromosome, and you only form the chromosomes during cell division. So the two strands of DNA are complementary. So one strand will fit with the complementary second strand, and they're going to run in an anti-parallel direction. So on one end, you've got the 5' end. It's going to be where you have a phosphate on carbon number 5. Here you have the 3' end. It's going to be a hydroxyl group on carbon number 3. Saying the 5' end and 3' end of DNA is like saying you read a sentence left to right. So here you're going to go 5' to 3' in one direction. The complementary strand runs in the opposite direction, and then you're going to have the complementary base pairs A and T, G and Z paired up for the rungs of the ladder. So if you were to separate this strand and have just this A, G, T, C sequence here, you could predict what is going to be the complementary sequence on the other side, because only one thing can possibly fit with it. So when DNA replicates, it uses this in what we call a semi-conservative model. Here in the gray, you have your two original strands. What's going to happen when you make copies of this to synthesize new DNA is you're going to pull these two original strands apart and use them as a template to make the second strand. So your new strand ends up being one part of the old, one part of the new that will make your new DNA. When this happens and you're separating this apart, you're going to have a replication fork that will form. And that's going to happen at the opening of the origin of replication. This enzyme helicase is going to come along and it's going to separate the DNA strands. It's going to pull them apart. When it does that, it's going to make an RNA primer. The RNA primer has to be synthesized, and as it goes along, the DNA polymerase will be able to join on there, and it will be able to synthesize your strand of DNA. So the term DNA polymerase, the ASE ending tells you it's an enzyme, and it's going to make a DNA polymer, is what a DNA polymerase is going to do. So it has to use that primer because DNA polymerase can only add on to existing strands. So when it does this, on one strand you're going to have the origin, and you're going to make your primer here. The origin was here in the center. The primer is going to be where you can start synthesizing, and DNA is going to be added on in the 5' to 3' direction. So here's your 5' end, and as you add on, you just keep going down to the 3' end. Being that it can only be done in that direction, we have a little bit of a challenge here when it comes to the second strand. So with this strand, what's going to happen is you start here at the origin of replication. It's going to jump back and read 5' to 3'. Then it has to jump ahead again and read backwards 5' to 3'. It will jump ahead again and read backwards 5' to 3'. So in doing this, we call this the leading strand here that's being synthesized continuously. It's all in one piece. This that has to be done in these small fragments called Okazaki fragments is the lagging strand. You have to make an RNA primer for each one, and later on these short stretches of Okazaki fragments are going to get joined together by the enzyme DNA ligase. So when we're making the copies of the DNA, when you go back here and you look, here where you have this RNA primer, you're not actually copying the DNA. You have this little region here that wouldn't get copied. So when you would go to make another copy of it, you're going to have this region here again that's going to eventually get pruned away and cut away into the gene. So to avoid this cutting away into the gene, we have telomeres that are going to be at the ends. So the enzyme telomerase is going to help to maintain the telomeres on the ends of chromosomes. Some chromosomes will have telomerase activity, some do not. So telomerase has an associated RNA that's going to complement the 3' overhang at the end of the chromosomes. So here you've got this 3' overhang. You're going to have the telomerase here that's going to help to fill it in and synthesize the complementary strand. So then the telomerase can shift and the process can be repeated. And finally, what you have is the primase and DNA polymerase that are going to synthesize the complementary DNA strand. So by having it be out here, you're going to buy a little bit of extra space. So this is a photograph of Elizabeth Blackburn, and she was the 2009 Nobel laureate. She was the scientist that discovered how telomerase works. Your telomerase activity is something that is still being studied quite a bit. It's one of the theories as to why we age, is that eventually we prune off all of the telomeres on our cells, and then they're no longer able to replicate without being dysfunctional. So when we're making copies of the DNA, there is always the potential for errors to be made. It's like if you have a paper that you're going to type into the computer, there's always going to be the potential for typos that will happen. So proofreading gets to happen with DNA polymerase. It's going to correct the errors during replication. So I think of the DNA polymerase as kind of acting like your autocorrect if you're typing something on your computer. The autocorrect is going to be good. It will pick up a lot of mistakes, but it doesn't catch all of them. And sometimes it will actually take words and put in the wrong thing. So it reduces the number of errors you have, but it does not eliminate them entirely. Some things you can end up with are mismatch base pairs. That mismatch repair will come along and it detects that the incorrectly added base is present after replication. The mismatch repair protein will detect this, remove it, and then the newly synthesized strand will have nuclease action, help to pull that out. Then there is a gap that's left behind, and it's going to be filled with the correct base pair. So with nucleotide excision, here is where you have thymine dimers. When UV light is going to be reacted with DNA, or DNA is exposed to the presence of DNA, you can have the thymines next to each other form a dimer. And they are no longer going to be read as two A's when they do that. So in normal cells, they get excised and replaced so that you don't end up with a mutation. So the central dogma of DNA states that DNA is going to encode RNA, and the RNA is going to in turn encode protein. This still holds fairly true for the most part. However, it is not absolutely the way things work 100% of the time. So occasionally we do just make the RNA, and we never turn that RNA into a protein. It just functions on its own as an RNA. So the process of making a protein from your DNA is going to begin with transcription. So here you've got your DNA template strand and the non-template strand that's complementary. The promoter is going to be a regulatory region ahead of the gene that you actually want to transcribe. So transcription begins when the DNA becomes unwound, and you're going to have a transcription bubble. Enzymes and other proteins are going to get involved and bind to the promoter at this section. So as you do that, it's going to go along. It's going to use an RNA polymerase enzyme this time, because you're going to make an RNA polymer from the DNA. So during elongation, you've got the RNA polymerase that's going to track along the DNA template. So it's going along this DNA template here. Here's your RNA polymerase. And it's going to synthesize the complementary mRNA in the 5' to 3' direction. So the DNA will unwind itself as the DNA is read, and then when it's done, it will rewind itself. So when it's doing this, you can have multiple polymerases transcribe a single bacterial gene, while numerous ribosomes can concurrently translate the mRNA transcripts into polypeptides. So this is a way that a specific protein can be made very rapidly in a bacterial cell. So essentially, you can think of the RNA that you're being made, your messenger RNA, as the working copy, is being made as you go. And if you're still in the process of making those copies, that's fine. The workers can start with those first steps of the instructions on the mRNA, well, you're still adding more of it on. So you can begin before you've actually made the entire RNA transcript. So we call this, when multiple ribosomes are hopping on there to follow the instructions, polyribosomes. So not only can you have multiple RNA transcripts being made, but you can also have multiple ribosomes that are reading that individual RNA transcript. They just move behind each other, reading one step at a time. When we make our primary RNA transcript, what comes out in a eukaryotic cell is not always working and ready to go. It has to be cleaned up a little bit. So we call the initial part the primary RNA transcript. It's got these regions called the exons and the introns. The introns are the intervening sequences that have to be spliced out. The exons are the expressed sequences. And then on the end, you're going to have to put some markers on there as well. So you have this primary RNA transcript that is made, and then it has to go through RNA processing. What you're going to do is you're going to put on a 5' cap on the end, and a poly A tail, which is just multiple adenines on the 3' end. You're going to pull out the introns and then attach those exons together. The protein synthesis machinery is going to be the ribosome. The ribosome is going to have large and small subunits. It's going to come in and it's going to read the mRNA. So here is your mRNA strand that is your instructions. The ribosome is on here. Here you have a tRNA. The tRNA is your transfer RNA. mRNA is your messenger RNA. What the tRNA is going to do is it's going to bring the amino acid down to meet the codon or the proper instructions on the mRNA. It will also help facilitate attaching that amino acid onto a growing polypeptide chain. When it does this, it's going to read these codons, and it's a genetic code that's in triplet. So we have 4 different base pairs, A, U, C, and G in RNA. In RNA, you're going to replace thymine with uracil. There are 20 different amino acids. So because there's only 20 different amino acids, and we have 3 letters for each code, it's going to be 4 times 4 times 4 possible combinations. That's going to give you 64 codons with only 20 amino acids. So some amino acids will have more than one codon. So when you go along to read this, where you're going to start reading is always at the first AUG. So if we go back here and we look at this DNA sequence here, what you're going to be doing is you're making your complementary RNA sequence here. Where you start reading right here is the first AUG. AUG may not be on the end. It may be in a ways that you have to look for. So here you have the AUG. Next you have CCG. So when we look at this codon table here, the way we know that AUG codes for methionine is the first base pair A is on the left, the second is on the top. That's going to bring you down to this box. The third is found on the right. When you draw the line over to that box, it will show you where the AUG is. It's going to code for the amino acid methionine, which is abbreviated as MAT. So that's going to be the first amino acid. The second one in this example here is CCG. So when we come back to this codon table, first letter C, second letter C, brings you down to this box. Third letter is G, come across, it's going to code for the amino acid proline, PRO. One more example here, the third codon is CAA. First letter C, second letter A, third letter A comes across. So here, this GIN is going to code for your third amino acid. So this codon table is not something people memorize. If you need to be able to read it, you would always be given a codon table to do that. So here it's showing it in action. Translation is going to begin at this first AUG. Here is your tRNA. The tRNA has a region here that is complementary called the anticodon. And then on the end, you've got this amino acid methionine attached. So as this comes down, the large ribosomal subunit will come down around it, and it will slide down to the next space. So in the next space, it's going to be reading UUU. So here we go, UUU. The anticodon on the tRNA is going to say AAA. It's going to be complementary and match up. Here it's going to have this amino acid attached to it. It's going to facilitate linking it to the methionine from the first one, and then it will move on to the next step and read the CGA, which complementary will be GCU, and have this ARG attached. So there's 3 spots on your ribosome. We have the A site. This is where you are accepting the amino acyl tRNA that comes in. So you're accepting the new tRNA. It's going to move, then slide into the P site here. This P site is where you're going to have the growing polypeptide. After you have attached those amino acids together to make your polypeptide, it will then slide into the E site, which is the exit site, where that tRNA will be ejected and it can go out and be reloaded to bring another amino acid down in the future. So in eukaryotic gene expression, it's regulated during transcription and RNA processing, which take place in the nucleus, and then as well during protein translation, which will occur in the cytoplasm. There are many steps for regulation to occur. You can also have post-translational modifications. So you can regulate things when you are actually making your mRNA in transcription. We have regulation and processing here where you're going to make your primary RNA transcript into your final mRNA. And then we can have regulation steps down here during translation. So one of the things that can happen is alternative splicing. There are 5 basic modes of alternative splicing where you have segments of the pre-mRNA with the exons, in this case shown in blue, red, orange, and pink, that can be spliced in a variety of ways to produce a variety of new mature mRNA sequences. So here you have exon skipping, where you're just going to pull these out, and you'll have 2 possible ones made. This one you just skipped one of them. Here you just used the first and the third. Here you used all 3. You can use mutually exclusive exons. So here you're going to have either 1, 2, and 3, or maybe 1, 3, and 4. Or this I guess would be 1, 2, and 4, or 1, 3, and 4. You can have alternative 5' donor sites, so it can start at different places. You can have alternative 3' acceptor sites, and then you can have intron retention, where you may actually keep a segment. So there are numerous other videos that you can watch in here that I strongly recommend watching these, and doing just small pieces at a time. You're going to have to spend a lot of time with this for it to make sense. It is not going to make sense off of just one, but the animations are very helpful in understanding this process.